More experiences equals a bigger brain by Rosenwig and Bennett, 1972. Rosenwig conducts a lab experiment to investigate the effects of deprived or enriched environment on neuroplasticity.
The independent variable is the condition that rats were placed in, either impoverished, enriched, or in a laboratory cage. The dependent variable were MRI scans of the rats' brains. The control group was the laboratory cage.
For the procedure, 3 male rats were chosen from each litter and were randomly assigned to one of the 3 conditions. These were impoverished, enriched, or lab cage. They used 12 rats in each of these 3 conditions for 16 experiments. For the standard lab cage, it had several rats in a regular sized space with food and water. In the impoverished environment, it was much smaller, was isolated in another room where the rat was alone with water and food. And for the enriched environment, there were 6 to 8 rats in a large furnished cage with toys to play with. The rats' brains were dissected and sections were measured, weighed, and analyzed after the rats lived in these environments between 4 and 10 weeks.
Cultural considerations. All labs were from the same litter. Gender. All male rats were used. Ethics. There could have been possible animal cruelty or mistreatment.
A few strands of the study and method include: changes in the brain due to experiments might lead to a better understanding of how memories are studied in the brain. Another strength includes the fact that the study provides information about correlation between malnutrition and intelligence. A third includes the examinations were done in random order by code, so that the person doing the autopsy would not know which condition the rat was in to avoid bias.
A few weaknesses of the study and method include: 1. The results from the use of rats may not be applicable to humans. 2. Lab experiments are always artificial. And 3. Handling may have been what caused variability in results and not the enriched environment.
There are 3 main results of the Rosenzweig study. One is the fact that the cerebral cortex of the rats in the enriched setting is heavier and thicker than the rats in the impoverished setting. Another result is that the enriched rats had larger neurons and greater ratio of RNA to DNA, along with more chemical activity. A third is the fact that the rats in the enriched setting, synapses are 50% larger than the rats in the impoverished setting.
In terms of the ecological validity, when the experiment was replicated, there was always the same results. However, the use of rats may not be applicable to humans, and the study should not be performed on humans due to ethical considerations. The lab setting also lowers the ecological validity because of high control over the results.
The results of the Rosenzweig study shows us that individuals in more desirable environments are more likely to have bigger brains. The cerebral cortex is associated with thinking, planning, and decision-making. The rats used in the experiment showed a thicker and heavier cerebral cortex, which means they are more likely to show more reasonable thinking, planning, and decision-making. This study helps us understand those who have trouble interacting socially. The study also helps us understand the correlation between malnutrition and intelligence. This is because malnutrition may make an individual unable to comprehend or consume new knowledge. The purpose of the Hyde study was to examine brain structure and behavioral changes in response to long-term music training in developing brains. The independent variable were the children who did or did not receive 15 months of instrumental music training. The dependent variable were the structural brain changes of the children. The control group were those who did not receive any music education.
Gathered information. The deformation automated morphometry or DBM scans on the children. The culture was 31 children as a mean of 6 years old. For ethics, it was approved by the Ethics Committee of Beth Israel Deacon's Medical Center. For gender, it was unspecified.
Some strains are that correlation between brain deformation measures and the melody arithmetic tests is consistent with functional brain mapping studies that have found activity changes using auditory musical regions. There is no behavioral or brain differences between the instrumental and control children at the baseline. All children are right-handed and are matched closely as possible in gender and age. All children had a DBM scan before and after the method.
Results of the Hyde study include the fact that instrumental children showed greater behavioral improvements over the 15 months on the finger motor test and the melody rhythmic test. Another result includes the fact that instrumental children showed areas of greater relative vowel size change over the 15 months as compared to controls in the motor brain areas such as the right percentile gyrus, corpus callosum, and right primary auditory region.
Brain regions are known to be critical importance in instrumental music performance and auditory processing. Results provide new evidence for training-induced structural brain plasticity in early childhood. Findings indicate that plasticity can occur in brain regions that control primary functions important for playing a musical instrument, and also in brain regions that might be responsible for the kind of multimodal sensory-motor interrogation likely to underlie instrumental learning.
In the Hyde study, children in a more musical environment psychologically benefit from increased brain function and density. Also, this study helps us understand if the efficacy of early start programs are really true and how starting your children earlier in education can help them later in life.